The research taking place in various fields each day is outstanding, from the latest medical news to the most out-of-this-world astronomical discoveries. There is plenty to get caught up on with the advancements being made. So today, here at Unexplained Mysteries, we will be taking a look at 3 new discoveries. Astronomers find 2 new galaxies hiding behind curtain of dust. Sometimes things are not always as they seem. A group of astronomers have been looking for a sample of distant galaxies, though their expectations had to be reassessed when they uncovered 2 seemingly invisible galaxies hidden behind what is being described as a curtain of dust, tucking the galaxies out of sight. The researchers used the Atacama Large Millimeter Array, or ALMA, to conduct their research, tracking, capturing and analysing radio waves. This is a less often used telescope, as typically the Hubble Space Telescope is capable enough, though as it uses ultraviolet and visible wavelengths, the dust curtain managed to stay hidden to Hubble. The galaxies have been given the ever-so-catchy names Rebels 12-2 and Rebels 29-2. It appears they are 2 of the most distant galaxies that we are aware of. The light from these distant galaxies travelled 13 billion years to make its way to Earth, though by now, as the universe has continued to expand, they are a whopping 29 billion light-years away. The study suggests that there could be plenty galaxies more tucked away and hidden just out of sight in the universe. This gives us lots of questions challenging our current understanding of the universe. The new research has estimated that between 10 and 20% of the galaxies from the early universe could now be concealed behind dust clouds. This study serves as a reminder to the benefits of moving away from the status quo. Had the research team used the Hubble Space Telescope, we could have remained unaware of these galaxies for decades, possibly even centuries. Alien organisms could hitch a ride on our spacecraft. We are in an exciting age of space exploration, with space companies worldwide expanding across more and more impressive horizons. Though as we continue our research, looking at exoplanets and searching for life, we could run the risk of encountering more and more problems too. Some scientists have begun to express concern that alien organisms could become an invasive species harming us here on Earth, or that we might be causing problems elsewhere, introducing our Earth organisms to alien environments. Think back to your science lessons at school. We know that ecosystems are fragile. Here on Earth, humans have caused problems beyond repair, letting organisms invade new environments they would not reach otherwise. One example of this is the South American fungus Austropurchinia seedi being introduced to Australia, where it stunted and threatened the eucalyptus trees native to Australia. Imagine if this happens on a much greater scale. We are not evolved and adapted to deal with alien biological threats of this kind. With Earth being in isolation by our nature as a planet, it is safe to assume the devastation would be even greater, having never encountered any organisms other than those that are our own. Could it be smart to stop trying to intentionally find and interact with alien life forms? Should we stop the search? While the idea of invasive species is frightening, scientists do acknowledge the risk of biological contamination like this is low, though with every mission out there, it does increase. Realistically, the changes of an organism surviving the intense conditions of outer space while clinging to the outside of one of our spaceships and making it back to Earth are minimal. The harsh environment does limit the possibility, though that does not mean we should not remain cautious. There are some regulations in place already to help ensure the safety of us here on Earth and any potential life on the planets we could be visiting. NASA and other big space agencies have long been aware of these risks, and so planetary protection policies have been in place since the 1960s, designed to look after all parties involved. Some scientists have expressed concern towards private space exploration, namely SpaceX. SpaceX's aim to travel to Mars with the SpaceX Starship program, coupled with their lack of agreement to adhere to the policies in place, all has presented a cause for concern. Researchers are suggesting an increase in the severity of the biosecurity protocols surrounding space travel. This includes a focus on detecting possible contaminants early on and having a series of procedures in place to respond to these threats quickly and efficiently. So, whatever you think about alien life out there, or are concerned about how finding alien life might go for us, there are plenty of plans, research, and theories in place. It does not stop the age of space exploration from being as exciting as it is. Slice of human brain is kept alive in a petri dish for 12 hours. A huge part of science we all want to advance in great leaps and bounds is, of course, medicine. A breakthrough study has seen scientists successfully keep a piece of human cortex alive in a petri dish for 12 hours, something that has never been done before and marks the potential for significant medical progress. Being able to observe and experiment with a 1 centimeter slice of human brain could lead to better drugs and treatments for diseases we know to be fatal. Emma Louise Louth, the lead researcher of the project, working at the University of Copenhagen, with the help of her team, was able to take quick action after taking a sample of the patient's cortex, making sure the tissue could survive. These steps involved the regulation of temperature and oxygen, cooling the sample down and making sure it stayed oxygenated, then submerging the tissue sample into a mixture of ions and minerals that was designed to mimic that of cerebral spinal fluid. This all was intended to keep the sample alive, ready for observation. In a press release, Louth revealed that this cortex brain sample stayed alive for 12 hours. That is 12 hours of research opportunity we have not had before. This complicated breakthrough has bridged a gap between animal studies to humans. Louth said in her statement, to borrow an analogy from another researcher, mouse studies versus human studies are basically like looking at a Nokia 3310 when trying to repair an iPhone. They have the same basic functions, but there is much greater complexity in the human brain. Animal studies have been a point of contention for years. Animals are simply different to us as humans. While the brain may work in a similar manner, how can we accurately determine that the processes within are the same between a human and a mouse? We already know that there are significant differences between the types of cells and receptors within humans and mice, so being able to use humans helps drastically. Louth and her team were able to look at the dopamine-enhanced connections between neurons in humans and mice. This means that the neurotransmitter we consider engaging with reward strengthens the connection between neurons in the human brain. In short, Louth and her team looked at how dopamine is different in humans than in mice. In practical terms, this has real-life application in terms of testing drugs. If we are able to keep these brain tissues alive long enough to observe effects, we will be able to see how the different species' neurotransmitters react differently to various drugs, meaning we know what will work best for humans, not mice. Of course, when studies begin to work with human brains, there are ethical considerations to be considered. And no, these concerns are not whether it is unethical to plonk humans in mazes and make them run for cheese. Is it okay to cut up, experiment on, and remove bits of a living human brain? Louth has assured the public that no pain is felt, no memories are stored in the tissue, and without all the other parts of the brain being connected together, there is no painful damage being inflicted on the sample. So, if we can perform these studies on emotionless brain samples, why would we continue to work with living animals? The opportunities this study presents in terms of both scientific validity and the ethics and well-being of animals is monumental, not to mention the realistic difference this could make to medical research. Louth believes this research could help rehabilitation after a stroke or other acute brain damage where synaptic connections are lost. So, what's next for this research? The University of Copenhagen scientists are now aiming to keep the sample alive for longer, with the next milestone being 10 days. It will not be long until this research begins to have a tangible impact upon medicine. But what do you make of these fascinating discoveries? Be sure to let us know your thoughts in the comment section below, and help us by growing this community while working to solve these unexplained mysteries. Thank you for watching, and don't forget to subscribe for more videos.